Greetings. So today we want to continue our discussion of the circulatory system. And in particular, we want to talk about the plumbing, that is, the vessels by which the blood is flowing through the body, and its delivery of oxygen to the tissues, and the removal of waste materials from those tissues. So you all know that the heart then is the pump for the circulatory system, and that these will be the vessels, and that the vessels plus the heart form a closed system. So our learning objectives will be to describe the function of the arteries, the arterioles, capillaries, venules, and veins. These are the various vessels through which the blood will be flowing. And secondly, we want to describe the relationship between pressure, flow, and resistance in regulating the distribution of the blood throughout the body. Third, we want to define the systolic, diastolic, and mean arterial pressures. And you've already heard about systolic pressure and diastolic pressure, which is simply systolic is the pressure generated by the heart when it's ejecting blood. And diastolic pressure is the pressure that's within the vessels as all the valves are closed and no blood is then leaving the heart and entering into the arterial system. And then fourth, we want to explain what's known as the myogenic response, active and reactive hyperemia, and then reflex control of the blood vessels' resistance. And then five, talk about the factors that will affect the transcapillary movement of fluids and solutes. This is the way that we deliver these fluids and solutes then to the tissues, and it is by local diffusion. And then six, we want to explain the differences between capacitance and distributing vessels and the relationship between the blood circulation and the lymphatic circulation. So we have a very large list of materials to cover, so let's get started. So the first here is to consider the actual circulatory system. And as I said, we have a heart, and the heart is essentially, as you know, composed of two separate hearts that are in series. The first delivers, that is the right ventricle, delivers blood to the lungs for oxygenation. And then oxygenated blood is returned to the left ventricle, and the left ventricle then distributes the blood to the rest of the body. As the blood is ejected from the left ventricle, it enters into a series of vessels which are called arteries. And these vessels, at the very beginning, the largest is the aorta. The aorta is what's called an elastic artery. This is an artery that has a lot of elastic tissue within its walls, and so it's able to stretch and then contract readily. These elastic arteries drain into what are called muscular arteries, and the muscular artery is simply an artery that has more muscle in its wall than elastin. And these muscular arteries are the arteries which will deliver the blood to specific organs. And the delivery of the blood to the specific organs is going to be in parallel. So when we deliver blood to the kidney, it's independent of delivery of the blood for instance, to the GI tract or to the lung. Once the blood is delivered to the specific organ, then it enters into the organ, and within the organ, the distribution within the organ will be governed by what are called arterioles. The arterioles are sort of the little spigots within the system, and they're the ones that determine which portions of the organ will be perfused. From the arteries, the blood drains into the capillaries, and this is the site where we have exchange of solutes and fluids, such as water, between the tissue and the bloodstream itself. And then draining from the capillaries are the smallest of the veins, the venous system, which is called the venules, and these drain into veins, and then the veins eventually return to the heart, return the blood to the heart through the vena cavas. The veins have the largest capacity within the system. So the most amount of blood is actually sitting in the veins at any one time. And something about 60 to 70% of the total blood that's within the system is on the vein side. In addition to that, the veins are a low pressure system, and so the veins have valves where the blood is able to move through this system in a unidirectional manner, but cannot move in the opposite direction because the valves prevent backflow through the system. So the veins have large capacity and low pressure. This is in contrast to the aorta and the elastic arteries and the muscular arteries, where this is a high-pressure system. So in this particular side, then, the pressure that's being generated by the heart is delivered into the aorta and then from the aorta into the arteries. And this will have a high-pressure system on that side. Okay, so what are the rules for the cardiovascular system? So the first rule is that the flow is going to, of blood is going to be unidirectional through the system, and that the flow is generated by pressure gradient, which is generated by the heart. And the flow will go from high to low pressure states. The second is that the resistance will oppose flow as the blood is moving through the vessels. There's a certain resistance to the blood flow, and this is due to the blood cells bumping up against the walls, the fluid bumping up against the walls, and the blood cells themselves bumping up against each other. So there's a type of resistance within the system. But the resistance is also dependent on the length of the tubes, which in the human is essentially fixed once you reach the adult state. And secondly, on the radius of the tube, and this is actually the most important, this is the control site. So if you change the radius of the tube, you can increase resistance within the tube if you make the radius smaller. So a smaller diameter vessel has a higher resistance. Excuse me, and we'll come back to that point later. And then the viscosity of the blood. And as you know, if you do blood doping, you can increase the viscosity of the blood. If you have dehydration, then the loss of volume from the vasculature can increase the viscosity of the blood. But these are unusual circumstances. And under normal circumstances, the viscosity of the blood is not a factor. The third thing that we have to think about is flow rate, and that is simply the volume of the blood that passes through a given point per unit time. And this is liters per minute, and this will be determined by the pressure gradients and by the resistance within an area. And last, we'll have the velocity of the blood, and this is how far the volume of blood travels per unit time. This is in millimeters per second. And this is determined predominantly by the cross-sectional area. The slowest flow, the slowest velocity or speed of flow through the system will be found in the capillaries. The capillaries are very small diameter vessels, but there are many, many, many capillaries. And so the total cross-sectional area of the capillaries is quite large. And so the flow then will be very slow across the capillaries. And this will be important because we need to have time for exchange of the solutes and fluids across the capillaries and with the tissues. And this, of course, is by diffusion. So we need some time for the diffusion to occur. So there'll be slow flow then through the capillaries. Okay, so let's look at the pressure gradients through the entire system. And we'll only consider the pressure gradients through the circulatory system that's delivering blood to the body. As you know, we have the blood, when it is ejected, it has a systolic pressure. And the systolic pressure is usually 120, or we'll consider it to be 120 millimeters of mercury. And that the diastolic pressure will be 80. And so when the blood is ejected, then we have a high pressure. But then as blood is not being ejected from the heart, the blood that's entering, the pressure then will drop, and it drops within the aorta. So we see then a pulsatility of the blood as it's going into the aorta, and that's between the systolic pressure and the diastolic pressure. And that pulsatility is maintained through the aorta and in through the elastic arteries and into the muscular arteries. This pulsatility is actually quite important. So what happens is that in the elastic arteries, as the blood is being delivered to them, and we have a systole, we have a high pressure, then the walls of the arteries are stretched because there's a lot of volume that's being delivered to the vessel. And then the stretching of these walls during diastole maintains the pressure gradient within the vessel, because as there's no blood being emitted from the heart, then the blood that's within the elastic artery recoil then is pressing against the fluid and maintaining that pressure gradient. This ability of the elastic artery to stretch to volume, it changes volume with a change in pressure, is called compliance. And so that's so compliance then is equal to a change in volume over a change in pressure. So the elastic arteries then have a set type of have a compliance which allows them to maintain the pressure gradient. As the blood then flows from the elastic arteries into the muscular arteries, and we begin to distribute it to the different organs, then the resistance within the system starts to cause a drop within the pressure. But the dominant area where the pressure drop occurs, it occurs within the arterioles. So the pressure then will fall dramatically within the arterioles, and at that time, the pulsatility will also be diminished as it's moving through the arterioles. So the arterioles then are our spigots. So the arterioles are the dominant way that the blood will be distributed within the organs, and that this distribution causes an increase in resistance to flow, and so we have a pressure drop, a dramatic pressure drop across the arterioles. Then from the arterioles, the blood will enter into the capillaries. And again, there's a pressure drop across the capillaries, but it's less than what we see through the arterioles. And the pulsatility then of the blood is essentially smooth. And once we get into the veins and into the vena cava, which is the largest of the veins that is delivering the blood back to the heart, then the blood that flow now is smooth, and we have a very low pressures within the system. So there's two things to remember. One is that we have pulsatility of blood flow on the arterial side, and that, secondly, that the blood pressure is going to diminish as it runs through the entire circuit and returns to the heart itself. And it's pretty easy to remember the idea about the pulsatility, and that is simply that if you cut your wrist and the blood is spurting out from your wrist, you know that you've cut the artery. But if you cut the wrist and the blood is just oozing out, then you know that you've cut the vein. Okay, so what about these blood pressures? So the blood pressure then we're referred to as the mean arterial pressure, and the mean arterial pressure is calculated by the diastolic pressure plus 1/2 of the systolic, 1/3 times the systolic pressure minus the diastolic pressure. And the systolic pressure minus the diastolic pressure is referred to as the pulse pressure. So why is it that the mean arterial pressure is not simply taking the systolic pressure plus the diastolic pressure and dividing by 2? Well, the reason for that is the equation is weighted for the diastolic pressure, and that's simply that the heart stays in diastole longer than it is in systole. And so the amount of time so the pressure, the diastolic pressure then has a larger component to calculation of the mean arterial pressure. Okay, so now once we have the blood is going to be distributed to the various organs, I told you that the distribution will occur through a parallel system, so that I can distribute blood to the GI tract and not be distributing blood, say, to the skeletal muscles. So why is it that we have to have this movement of blood from one organ system to another organ system depending on demand? And that's simply because there's not enough blood within the circulatory system to allow us to perfuse all of the capillary beds within the body at one time. If all of the capillary beds were dilated so that they were all open, so you were able to perfuse every capillary in the body, then you would pass out. So in order then to to distribute the blood to specific organs that need the blood for that particular state, then we have these distributing vessels, which are called the arteries. So the flow, as it's going through this system, will be dependent on the pressure gradient divided by the resistance within the system. So for the entire cardiovascular system, the mean arterial pressure minus the pressure in the vena cava divided by the total peripheral resistance is what we will consider. So that's at the if we're talking about the entire system. But within the organ itself, it's it would be the pressure that's within the mean arterial pressure minus the venous pressure of that organ divided by the resistance of that organ. And in both the case of the entire systemic circulation as well as in the case of the organ, the venous pressure is very low compared to the mean arterial pressure, and so it's considered to be essentially zero. The vena cava has a pressure of about 12, where the mean arterial pressure will be something like 100. So we really don't factor it in and sort of make the adjustment that the cardiac output then is equal to mean arterial pressure divided by the total peripheral resistance, or the perfusion pressure for the organ is equal to mean arterial pressure divided by the resistance in the organ. Now, once we're within the organs, then specific capillary beds will be perfused within the organs. One other point that I should make, and that is that although the arteries are regulating which blood flow goes to particular tissues at any one time, for instance, in the fed state, the GI tract is getting a large amount of the blood flow, but the skeletal muscle may be only getting 20% of the cardiac output. The two organ systems that always get 100% of their needs, and that is the heart and the brain. So the heart and the brain, regardless of the state, whether it's a fed state and we're diverting blood to the GI tract, or it's a state of exercise where we are diverting blood predominantly to the skeletal muscle and not so much to the GI tract, the heart and the brain get 100% perfusion. Okay, so let's talk about arterioles. So arterioles then regulate the blood flow within the organs. So in the arterioles have a complicated set of control systems. The first is that the arterioles are going to be governed by local conditions, and that is, and they are essentially three types of local conditions. So the first is the myogenic response, and this is simply that if there's more volume, more pressure that's being delivered to the arterioles, then the arterial walls, the smooth muscle that's within their walls will stretch. And when they stretch, that'll cause a contraction, because you're opening the stretch-activated calcium channels. And then these smooth muscle cells will contract and restore the original diameter to the vessel, or perhaps even make it smaller. So this will be a change in wall tension, and that change in wall tension maintains the flow rate or pressure within that system. So that's a simple myogenic response. The second is called hyperemia, and this is when there is a change in the interstitial space that's the components within that interstitial space outside of a given tissue. So for instance, if we're exercising and our skeletal muscle is using up a lot of oxygen and is generating a lot of CO2, CO2 levels within the interstitial space outside of the outside of the skeletal muscle will rise. In addition, you can have an increase in protons within that area, or with potassium within that area. And this causes increase in these metabolites cause an active hyperemia where the local arteriole will dilate. So this causes the smooth muscle then to relax in the walls of those arterioles, and more blood is fed into those very active sites. This effectively washes away the byproducts, these metabolites that were generated by contraction or by the activity of the skeletal muscle. This is, so this is called active hyperemia, because the muscle is actively working and it is generating these metabolites. There's a second type of hyperemia where, again, we will have an opening of the, we have relaxation of the smooth muscle of the arteriole and an opening of those vessels and allowing more perfusion of the capillary beds in that region. And this is due now not to an active exercise state, but in fact to the loss of perfusion within that area. So if for some reason there's ischemia within a specific region of the organ, then metabolites will accumulate within that area. So the metabolism of the cells is continuing. They're using up oxygen, but new oxygen is not being delivered to the cells, and local levels of CO2 will rise. When you open the perfusion to that region after the ischemic event, then you have a greater flow of blood through the region, and that's due to this reactive hyperemia. So when does this occur? So you all know if you go into the doctor's office and they're going to take your blood pressure, they put a blood pressure cuff on your arm, and then increase the pressure outside of the artery that's perfusing this is the brachial artery that's perfusing your forearm. They will collapse the brachial artery, and when they do so, then the blood flow to the forearm is stopped, and you now have an ischemic state for the forearm. Then, as they slowly release the pressure of the cuff, so as they're releasing the pressure on the cuff, blood flow will resume within the forearm, and at that point, all of these arterioles and the arteries in this area will dilate and allow more blood perfusion to occur transiently. And this is washing out local metabolites that had increased during the ischemic state. Now, the arteries and arterioles can also be regulated through reflex control. And in particular, we're talking about arterioles. The reflex control, the first is the sympathetic innervation. And sympathetic innervation, norepinephrine is the neurotransmitter. It will work on the alpha-1 adrenergic receptors, which are present on the smooth muscle of these arterioles. Secondly, we can have hormones which will act in this area, and the hormones then can be either constrictors or dilators. And so the hormones can cause the smooth muscle to either relax if it's dilation, or to constrict if it's or to contract if it's constrictors. And in the case of vasopressin or antidiuretic hormone, this is a constrictor, and epinephrine is again a constrictor. It acts on the alpha-1 adrenergic receptors. Now, there are some cases where epinephrine can act on the beta-2 receptors, and in that case, the beta-2 receptors, for instance, on the bronchi, the smooth muscle of the bronchi, the epinephrine will cause a dilation, a relaxation of those smooth muscles, a dilation of the bronchi, and that the tubes, the airways, will increase in diameter. So depending upon the type of receptor that the epinephrine is acting upon, you can either have constriction, which would be an alpha-1 adrenergic receptor, which is on the blood vessels, or the beta-2 adrenergic receptors, which are found on the bronchi, the smooth muscle of the bronchi. Okay, so these arterioles then are acting as nozzles within our vasculature, and the dynamic change within their contracted state, that is going from their basal tonal state to either contracted or relaxed, changes the diameter of the arteriole itself, and so the blood flow through that vessel will be affected. It not only affects the blood flow through the arteriole, but it affects the blood flow in the capillary bed, which is downstream, immediately downstream by that arteriole. And that's what's diagrammed here. So on the y-axis, we have pressure, and pressure is increasing in this direction. And along the x-axis, we've diagrammed artery, arteriole, and the capillary. And what's shown is the change in the arteriole. So if we start with looking here at at the blue or the basal state, this is our basal tone for the smooth muscle in the walls of the arteriole. We see that we have a drop in pressure as the blood is flowing across the arteriole. And this drop in pressure is due to the resistance. It's a small, narrow tube, and so it has high resistance as the blood is flowing through the tube. If we cause a relaxation, if we cause a relaxation in the smooth muscle that's in the walls of the arterioles, then the blood flow within that state then increases, and that means that downstream in the capillary, the pressure now is increased. There's more flow delivered to the capillary, and so there's more pressure now, a higher pressure within the capillary. And conversely, if we constrict the arteriole and close the lumen of the arteriole further, then the pressure falls within the capillary. There's less flow delivered to the capillary bed, and so therefore less flow, less pressure within the capillary bed, immediately downstream to the capillary. One of the ways that you can think about this system is that the arteriole is the nozzle that's on your hose, and that if you open the nozzle completely or close the nozzle, it changes the flow rate to the tube, the actual hose that's attached to their nozzle. And that hose that's attached to it would be the capillaries. And if you think of the capillaries as a soaker hose, which are full of little holes, then you'll understand that as the flow goes through the capillary, that the flow rate then is slowed as it goes through the capillary, and that there's much interaction between the fluid within the capillary and the outside. So there's essentially no barrier between the movement of the fluid and the garden itself. All right, so the pressure gradients that are found within the capillaries are actually very important for delivery of solutes and fluids to the tissues that capillary bed is perfusing. And that's what's diagrammed here. And we have effectively two pressures that we need to consider. So the first pressure is simply that the hydrostatic pressure. So this is the mean arterial pressure that's coming into the organ. So it's a hydrostatic pressure coming into the arteriole. And the hydrostatic pressure, as it goes across the capillary, it falls, such that the pressure that exits the capillary is less than the pressure that entered at the arterial side. And that's due to the resistance within the flow of the capillary vessel itself. So that's our hydrostatic pressure. And the second pressure is that which is generated by the oncotic pressure, which is generated by the proteins which are within blood. And they're attracting water or keeping water within the system. And so the oncotic pressure, as it moves through this region, the oncotic pressure is essentially stable. So we have hydrostatic pressure and oncotic pressure. And on the arterial side of the capillary, as you see, the hydrostatic pressure is greater than the oncotic pressure. And when the hydrostatic pressure is greater than the oncotic pressure, then there is a net filtration. That is a net movement of fluid from the capillary itself across the capillary walls and into the interstitial space. And this occurs in most regions of the body. There's a few exceptions where you have sealed capillaries, but under most conditions, then there's an easy movement of materials from the capillary across the endothelial cells and into the interstitium. On the opposite side, on the side towards the venule, now we have the hydrostatic pressure is less than the oncotic pressure. And under these conditions, then fluid is attracted back into the capillary. And that's due to the proteins which are present within the blood. And this is predominantly albumin. So under these conditions, we have reabsorption. So reabsorption then occurs when the oncotic pressure is greater than the hydrostatic pressure. And this is under normal conditions. So what happens under a condition where we have very high pressures within the system? So let's say we have hypertension within the system, and now instead of having a blood pressure of 180 over 120 over 80, we may have a blood pressure that's 180 over 90. Under these conditions, so hypertension, under hypertension, the hydrostatic pressure, as it's going across the capillary, stays relatively high. And the oncotic pressure is the same as it was before. But if you notice now, all the way across the capillary, from the arterial side to the venule side, we have a hydrostatic pressure which is greater than the oncotic pressure. So under these conditions, we will get filtration. And you get filtration across the entire capillary, including on the venule side. And there is no movement of fluid back into the capillary toward the venule side. So there's a net movement of fluid out into the interstitium or out into the tissues. And under this condition, we then have a pooling of fluid outside of the vasculature, and this is called edema. So the pressure gradients across the capillaries determine whether or not we have a net filtration and net reabsorption, or whether we have only filtration excuse me, across the entire capillary. And we can also have a condition where we have hypertension, and under hypotension, then we'll get the opposite. And under hypotension, we can have a situation where all the way along in our capillary, the tension that's generated, the hydrostatic pressure that's generated is very low. So if we have a very low hydrostatic pressure, so we have a hypotensive situation, then the hydrostatic pressure starts very low and drops across the capillary. But under these conditions, the oncotic pressure now can be, in fact, higher. And as the oncotic pressure is greater than the hydrostatic pressure, then there's reabsorption of fluid back into the capillary all the way along the capillary bed. So the pressures then that are feeding the capillary bed are important for the delivery of oxygen, nutrients, and so forth to the tissues. And then, in addition, they're very important for the removal of fluids. Once we've delivered these materials to the tissues, then the fluid needs to be reabsorbed and re-enter into the circulatory system. What about this extra fluid? So there's sometimes there's a mismatch in the type of this fluid. So the fluid is leaving at the arterial end, but not all of it is taken back up into the venal end. And there's a little bit of extra fluid then that stays out into the tissues. This fluid is picked up by another circulatory system, which is called the lymphatic system. And that's what's diagrammed here. The lymphatic system is a system that starts as a blind-ended capillary in the tissues themselves, and then drains the fluid from the tissues, this extra fluid, this lymph, from the tissues, and returns it to the bloodstream. And it returns it on the venal side, near the vena cava, back into the circulatory system. En route, it'll go through one filter, and these filters are called, or more, and these filters are called lymph nodes. On a given day, you can have as much as 3 liters of lymph returned to the blood. So this is actually a fairly significant amount of fluid that needs to be returned from the tissues back into the circulatory system. The lymphatic drainage is found in almost all parts of the body, but it is not present in the CNS. And disruption to this lymphatic system can have profound effects on edema within the tissue. So, for instance, in individuals who have breast cancer, often these nodes, these lymph nodes, are removed from the draining lymph nodes are removed, which are underneath in the armpit, which is near the breast that was affected. And these nodes are tested to see whether or not there's metastasis of cells that has occurred, whether or not there's cancer cells within these little filtering units or nodes. When they strip these nodes, and also possibly nodes that are further within the system, and disrupt the vessels, then fluid is no longer able to be drained away through the lymphatic system and back into the circulatory system. And so fluid can build up within the lymphatic drainage. And then to get the fluid back into the body, then an individual has to massage the arms or exercise the arm to help move the fluid back through these vessels and back into the circulatory system. All right, what's our general concepts? So the general concept, the first, is that the heart consists of two pumps, and that drive the unidirectional flow of blood through the pulmonary and the systemic circulations. Secondly, that this vascular system is a conduit for the blood flow, and it's a dynamic system because it can control the distribution of the blood from one area of the body to another. And that the distribution to the organs is in parallel, so that if I shut off the distribution to one arm, it does not affect the flow that's going into the other arm. So this arm is not in series with this arm. So these arms are being perfused in parallel. So I can affect the distribution of blood here and not be affecting the distribution of blood over here. Thirdly, the arteries are low-resistance conduits that maintain the pressure, and particularly the aorta and the elastic arteries. And they maintain the pressure then drained diastole when there's no flow of blood coming from the heart, and allow this pressure gradient to be maintained so that we can have distribution of blood to the organs. Fourth, the arterioles are our dominant site of resistance within the system, and that they determine what is the region within the organ that will be perfused. 5, capillaries are the site of exchange, and it's the balance between the hydrostatic and oncotic forces that determine the direction of fluid movement, either into or out of the capillaries. So when we have a higher pressure than, or hydrostatic pressure is greater than oncotic pressure, there's a filtration and there's a net movement of fluid from the lumen of the capillary into the tissue. But if the oncotic pressure is greater than the hydrostatic pressure that's present within the tissue, then the fluid will move from the tissue back into the bloodstream. And 6, veins are low-resistant conduits for venous return, and they are volume reservoirs. That is, they have a very large capacity. So about 60 to 70% of the blood within the system at any time resides on the venous side, or on the vein side, the venous side. 6, the sympathetic nervous system constricts the smooth muscle of the vein and can increase the venous return and thereby increase stroke volume and cardiac output. And we know that is called the preload. So we can increase preload by affecting the venous return. 7, the lymphatic system provides a one-way route for the return of interstitial fluid to the cardiovascular system. And this interstitial fluid is called lymph, and it is the sort of little mismatch between what's being filtered across the capillary and what's being reabsorbed back into the capillary. And 8, disease states that alter the hydrostatic or oncotic pressure can result in edema. So if we have hypertension, they can generate edema by having the hydrostatic pressure high all the way across the capillary. And so we're filtering all the way across the capillary. There are other states where the oncotic pressure can be reduced within the system, such as if you have a problem with the liver. Let's say you have cirrhosis of the liver, and under these conditions, then the liver is not able to make sufficient quantities of albumin. So the oncotic pressure within the blood is less. And that means that the attraction of water back into the bloodstream is less on the venous side. And so we will have a higher hydrostatic pressure than oncotic pressure all the way across the capillary. And again, edema will form. You can have problems then during heart failure where the hydrostatic pressure is changed, liver disease, as I've just described, where the oncotic pressure is changed, or in kidney diseases where we are holding too much water or we're not able to urinate, and so there's a large volume within the system, so there's a high pressures within the system, or we have protein malnutrition where again the oncotic pressures can be diminished. So the system then is a balance between the hydrostatic pressure and the oncotic pressure, and fluids will be distributed accordingly as to which pressures are the highest across the capillaries within the tissues. Okay, so see you next time, and we'll talk about how this entire system is a gigantic reflex loop and how the reflex loop is controlled. See you then.